Okay, children. So we shall start with the new part, that is the pest-resistant plants. Okay, in the previous class, that is in yesterday's class, we had done the BT plants, and BT, as I said, was the gene which is derived from the Bacillus thuringiensis, which produce the protoxin, and the protoxin, that gene, when introduced into the embryo of any of the plant, then all the cells will have this gene, and all the cells will produce those protoxin, and all the cells will be able to produce that immunity against those type of bacteria, those type of insects. Okay, so that we are done. Now today we are going to study a new thing, that is the pest resistant plants with the RNA interference. Okay, now what is RNA interference? We know that RNA, that is the central dogma. Central dogma states that DNA make RNA and RNA make protein. The central dogma says the DNA make RNA and RNA make proteins. The proteins can work as an enzyme, and enzyme carry out a particular reaction. Now DNA is a double stranded, RNA is single stranded, not necessarily helical, it can be of any shape, no problem. Okay, if we have an ability to make the anti chain, that is, this is sense, this RNA, since it is going to make a protein, so we will call it as a sense RNA. And if we have got antisense RNA, say for example, its sequence is AUU, and suppose we got another strand which we add from outside, which have UAAGCCGUA, so it will be just complementary to it, and no sooner the complementary DNA RNA is added, they will join together to form a double stranded RNA. RNA, of course, is always a single stranded. Now we cannot make it as a helical. Sorry, I am wrong here. RNA is not helical, but whatever. So it is a double stranded. It is a double stranded RNA. So when it is a double stranded RNA, how will it express its function? Because to RNA to work, it has to combine with the ribosome, and on the ribosome, the exposed nitrogen base nitrogen bases are read in the form of codon, in the form of codons, 3 set में. ठीक है? Now since there is nothing to be exposed, and when there is nothing exposed, how can it function? So this particular strategy is used for RNA interference. Okay, let us see what it's saying. Pest resistant plant. Several nematode parasite. We already know nematodes are most of the nematodes are parasite, and they are parasite even to the plant. Okay, so several nematodes are parasite to a wide variety of plants and animals, including human beings. So we are not talking about the animals and human beings. We are talking about the plant. A nematode, name is Melaignae incognita, infects the roots of tobacco plants. Okay, now it causes a great reduction in the yield. The productivity of the tobacco is greatly reduced because of this type of infection. You can see the next page, the diagram is here. Here you see when the nematodes are there, the appearance, their roots become something like this, and their ability to absorb nutrients greatly decreases. Okay, so a normal root should be like this. So now what we are doing, now what we have to do here, a novel strategy was adapted to prevent the infestation, which was based on the process of RNA interference. RNA interference. Another RNA which will interfere its function. Okay. So RNA interference take place in all the eukaryotic organisms. The method of cellular defense. Eukaryotic organisms have this. Okay, up to a certain extent. Okay. This method involves silencing of specific mRNA due to the complementary double-stranded molecule that binds and prevents translation of mRNA, that is silencing. So this is a novel thing which we are seeing. That is, the RNA is there, but RNA is not able to function. It is something like that. If a most notorious person is there who used to cause all type of this thing, problem in the class, and suppose somebody comes and just punishes him and asks him just remain here only, you will not move from here, and the class will be just silent, quiet. The same way, the RNA's function is just made quiet, and that is called silencing of RNA. Okay, this method involves silencing of specific mRNA due to the complementary double-stranded molecule that binds and prevents the translation of mRNA, that is silencing. The source of complementary RNA could be from infection by virus having RNA genome or mobile genetic element, that is transforms, that replicate via RNA intermediate. तो हमको कहीं से लेकर के आना है. Now the source can be, these type two sources are written, that is, it can be from the virus or it can be mobile genetic elements. ठीक है, तो ये है. Now using कहाँ से? Now the problem is that the nematoda is living there. You had to insert that nematoda is infected the roots of the plant, and we have to insert these RNA into the plant cell. How will we insert it? तो उसके लिए हमने क्या किया? We have caught that vector, that is Agrobacterium tumefaciens. You remember Agrobacterium tumefaciens, which we have studied in the previous chapter. That has a tDNA. The tDNA was responsible for causing tumor. So what we had done, that the tumor causing, the harm causing part is removed. Okay, and that tDNA still has an ability to enter into the plant cell. That this is the root of the a plant cell, and when this thing Agrobacterium tumefaciens attacks or comes here, it sends only the RNA, some DNA segments. Okay, and what is this DNA? That is tDNA, where the T stand for tumor. That is, it is tumor causing DNA, and as a result, what will happen? It will form a tumor, and the tumor will have specific cells which will secrete a particular type of substance, which is the food for these bacteria. तो हमने क्या किया? tDNA में ये ability है which can enter in the plant cell. What we have to do is we have to remove those harmful segments which causes tumor, but it will still have an ability to enter. उसके पास एड्रेस तो है कहाँ जाने का है, लेकिन उसके पास मैसेज नहीं है क्या डिलीवर करना है. So it will enter, but it will not cause tumor. That tumor causing segment is removed or it is denatured, and बाकी segment को अपन ने रख दिया. उसके साथ में we have inserted this RNA interference segment causing DNA, a DNA which will be producing this type of RNA. उसको इसके साथ जोड़ दिया. ठीक है. So now when it enters, it will produce an antisense of this sense RNA. So antisense is like this. So whenever sense and antisense RNA will come together, they will form double-stranded RNA, and the process will be stopped. And some of their vital processes, may it be respiration, may it be nutrition, may it be some other processes, when those will be stopped, the nematode will no more be able to survive. And all the nematode are died within a short span. You can see here that after application, 5 days after deliberate infection of nematode, but protected through novel mechanism. Within 5 days, the scene is entirely different. Okay, so I think it is clear to you how the mechanism of antisense RNA, which is sent onto the sense RNA, and they combine together and form double-stranded RNA, prevents the functioning, and that nematodes or any other parasite, we can use it for the many other parasites even. And this was first used for that, so that is been described in our book. Not all the things can be described in our book because we are just studying the same topic of each area. So ये है. So this is how this thing is used. बस आप ये RNA silencing याद रखिए. You remember that is what is called RNA interference, or how they are produced and how why they are, that is how their functioning are stopped. ठीक है. So ये सारा अपन को यहाँ पे जानना है. अब अपन पढ़ेंगे बायोटेक्नोलॉजिकल एप्लीकेशन इन द फील्ड ऑफ मेडिसिन. See, most of the biological activities are controlled by some organic molecule, and we cannot produce those organic molecules in the industries, in the factories, in the laboratories. ठीक है. Like the first example is given that yes. Yes.
सर वो एक छोटा सा डाउट था.
हाँ, छोटा बड़ा पूछो. एक मिनट में इसको,
सर हमने वो
1 minute, otherwise your this thing will come there. Okay, so I said that there are number of diseases caused in us because due to the lack of certain type of enzymes, or due to the lack of certain type of hormones, or due to some other type of lack of certain type of biomolecules. So if these biomolecules or enzymes or hormones, either we have to rectify the gene, which is very difficult because we can rectify gene in 1 cell, or we can rectify gene in 2 cell or 5 cell or 10 cell, but we cannot rectify genes in all the cells which are found in the pancreas, beta cells of Langerhans, which will producing insulin, or a particular type of organ. Suppose pepsin is not made, or suppose ADA is not made. So ADA are produced in the what you call in the WBCs. Lymphocytes are made in the bone marrow. Now you cannot replace all the cells of the bone marrow, okay, which are producing wrong type of this thing, and you send the correct gene. No, it is not possible. So what we have an alternative is that you produce these chemicals out somewhere and that you insert in them. So that is what we are doing in the genetic engineering. In the previous chapter, we have produced them in the bioreactor, and then that has been inserted in the form of injections or in the form of some other form. But here we are having little different strategy. ओके, तो हम वो वाला भी लगाएंगे और दूसरा स्ट्रेटजी भी लगाएंगे. नाउ आई डोंट थिंक देयर इज इनफ टाइम टू स्टार्ट a new topic, but I'll just give you an idea. That is, first we'll study about genetically engineering insulin, or rather I'll just read this thing. The recombinant DNA technology processes have made immense impact onto the areas of healthcare by enabling mass production of safe and more effective therapeutic drugs. Safe is a very important word. The substance which you are produced earlier, they say when the insulin was not produced in the industries, so the insulin we used to get from some other animals, but those insulin were not safe. They used to cause some, or trigger some type of allergic responses. Okay, so that were not safe. तो वो जो immunological और allergic responses हो रहे हैं, that type of chemicals we should not use. Okay, so it should be exactly similar, exactly same in working to the body's own condition. So that is very important. So further, the recombinant therapeutic do not induce unwanted immunological responses as is common in case of similar products isolated from non-human source. Again, at present, about 30 recombinant therapeutic have been approved for human use world over. The book is very old. You should consider this, that the book is very old. And when the book was written, rather when the data was collected for writing this book, that time 30 recombinant therapeutics, that is the medicines which are produced artificially in the industries by using biotechnological application, was only 30. Today there may be many, okay? And that 30 is world over, not in India. In India, only 12 of these are presently being marketed. Okay, and these are keep on changing. Sometimes we find that there is some other problem caused by some type of substances. Then we stop the use of those things and we start using something other. Sometimes because of the economy or some other reason, जो है we can keep changing. So this 12 and 30, the number which are written in the book, they are old data. However, when the question are asked, you will have to write these only as per book. लिख दो NCERT to be in the safer side. ठीक है? Because these things you cannot make a क्या कहते हैं हार्ड लाइन कि 30 ही होंगे या 12 ही होंगे, ये तो चेंज हो जाते हैं एनी टाइम. ठीक है? So that's all children for today. In tomorrow's class we'll study the next part. Thank you.